really glad that you talked about tools, because that was the next thing I wanted to ask you about. What is the state of the art in recording the neural activity associated with smell and decoding those recordings? What are the modalities for getting the data? How well can we do reconstructing what an animal is smelling?
Yeah, I mean, I'm happy to start, and then Dima continue. So the tools that we use to study neural activity in the mouse brain, let's say, and to study smells is not so different than for other systems neuroscience questions. We can use imaging tools to look at individual neurons. We can use electrophysiological recordings. So the differences are a little bit probably on the subtle side. Imaging, we have the advantage that the main structure we're interested in is actually superficial. We don't have to kind of go through large parts of the brain to actually access those neurons, which also means we can not only record, but we can also stimulate selectively individual regions or even down to individual cells quite effectively. On the electrophysiological side, maybe for using traditional electrical recording techniques, patch recordings, so very targeted recordings from individual cells quite well because the structure is quite superficial. Doing extracellular recordings is maybe a bit more tricky in the olfactory bulb to do on a large scale than it is in other regions, because traditional high channel count recording techniques rely on silicon probes where most of the recording sites sit along one or a few sort of linear axes, whereas most of our cells we are interested in studying the population of the output sit in sort of a sheath. So recording techniques like the ones developed at Paradromics, or what my lab has been working on for a while, where you have bundles of wires, are more effective to capture a large number of those output neurons. I think what we are lacking in terms of ability to understand recordings compared to other brain regions is probably a good understanding of the composition of different neurons. In most neocortical areas, people understand quite well. There are some statin positive interneurons, there are parvalbumin neurons, there are different types of pyramidal neurons, and we don't have very good genetic markers or understanding what these different cell types are doing. So we generally tend to lump together different cell types, different cells that probably correspond to different cell types and possibly different roles at this point. It's only slowly that we start getting a handle on what types of cells are there, which will help us to mechanistically understand what's going on.
I'm very much looking for the big screen of a large number of receptors for many others. And here I actually would like to let Gabe talk about knowledge that may be developed for that, because it's a new alternative technology that is, when we're doing imaging, we limiting ourselves to the superficial part of the bulb, and we can record activity of the top layer that is actually glomerular layer, that is receptor layer, or the very first processing layer. But Gabe is proposing, and what we kind of developing through the through the company, and together we're developing the alternative way to measure large number of receptor activity.
We're basically taking an electrode-based approach where we're using a electrode array to measure local field potentials. So we have a grid of electrodes. We're trying to get the pitch of those electrodes to a point where we're able to have singular glomerular resolution, and we can watch, spatially and temporally, the signals coming into the olfactory bulb, right? So we're intercepting it at a point before a lot of this processing takes place, and those signals get filtered and sent to the brain. So it's a way of looking at the outside of the olfactory bulb. We're doing it electrically, but there's optical techniques as well, well-known techniques, calcium imaging, which gives you fantastic spatial resolution to record those signals. From our perspective, the two most important tools that we're using are calcium imaging and doing electrode recordings using arrays, micro-ECOG type arrays.
Just to add to give, so the very interesting aspect of the grids that we almost hoping at one moment to wrap almost all the bulb. I don't think we can wrap all the whole bulb, but almost all the bulb in these grids and collect the information that will be extremely hard to get to any imaging recording. This is why I I'm so excited about this technology that we're developing with the game.
I wanted to ask in terms of the coverage of the bulb and odor space that we were talking about earlier. What do you feel like you can cover all of odor space, and do you think you have to?
Dima, we've had a lot of conversations about this, and it's very key to how we're going to develop our technology. Dima, do you want to tackle it?
So we started with 64 electrodes on the surface of the bulb, and we were able to detect some odors, discriminate some odors, but it's definitely not sufficient to get a comprehensive picture of the odor space. So imagine if you can get an electrode, surface electrode, that wrap all the bulb, maybe 60% of the bulb. We can put it on the lateral side, on the dorsal side. It really will require a fantastic surgical skills, but it's in the realm of possibilities. Now the technology became available to collect so much electrophysiological data, and we probably wouldn't reach full coverage, wrap completely whole bulb, but to access a large portion of the bulb will be quite possible. Now, if we can do it, we can also start playing different tricks. For example, if we're missing some specific receptors under our electrode, our genetic tools will allow us to move receptors under the electrode array. And the work of my colleagues who are factory geneticists already proved a possibility. They're working together with the laboratory of Tom Boza, who can overexpress specific receptor on the dorsal part of the bulb, or move one receptor to position of different locus. And a combination of genetic manipulation and novel electrode technology is just open amazing opportunities.
It's not inconceivable to even reach sort of the ventral side of the olfactory bulb, the output neurons with free photon imaging, or a combination of ultrasound imaging, etc. Not for a BCI or BMI, but from a research perspective. It's the advantage is the structure we're looking at is indeed really directly under the skull, and its size is on the order of maybe 2 millimeters or so. We would have to go to 2.5 millimeters. So it's still difficult, but the next couple of years might make it possible to access the entire olfactory bulb, if there's a good scientific driving questions to do it.
If we had a microwire technique or nanowire technique where we could use something penetrating that was of sufficient scale, we might be able to get access to those glomerular-like complexes on the ventral side. But with the technology that we're developing, we don't plan to get access because of that innervation that's coming through the cribriform plate up into the bottom, the ventral side of the bulb. If we try to cover anything around it, we would sever those connections. So we're trying to get as much of the bulb covered as possible using an electrical technique.